Good afternoon. Sorry, we'll try that again. Good afternoon. Hopefully everyone's retinas have recovered. I know that Dean Zadnick was getting a lot of questions for her clinic. Want to recognize a lot of leaders that are here today, a lot of friends, a lot of colleagues, great faculty, great staff. A couple of our deans here. Dean Bradford, wave if you don't mind. Dean Kreutz, I saw Dean Kreutz here. Other deans, Dr. Johnson's here from the CTSA. A great crowd and really representing the breadth of research happening across Ohio State each and every day. So welcome to you all to the 2024 Research and Innovation Showcase. This is one of the highlights of all of our years, and it's great to have everybody here. What a great way to start the showcase, inspiring video that really highlights the great work that you all are doing in your laboratories every day across the areas of the Enterprise for Research, Innovation, and Knowledge. But really, we all know that the research happens in the colleges and the labs and, again, the institutes that do the great work. What a great year it has been across Ohio State. First, we saw pictures of Professor Agostini. What a great tribute to the science that's happening every day. A highlight, I think, for all of us. We hear a lot of great things about athletics. We hear a lot of great things about Intel coming, but to have one of our own win the Nobel Prize, I know I got chills, and hopefully you all had an opportunity to interact with Pierre while he was here. It's been wonderful to have him on campus, and saw him just yesterday, and he is incredibly thrilled and so humble to have an award winner from Ohio State. That's not the only thing we've celebrated. In May, we opened the first building in Carmenton, our 300-plus acre innovation district that we sit in today. Today, teams of researchers in this new building are obviously working in the same labs across disciplines to find solutions, as we saw in the video, some of the most important and complex questions that our world is facing today. And to meet researchers where they are, our ERIC innovation and commercialization teams have actually now all moved out to Carmenton to really interact with researchers in the here and now to make it more accessible to our research community and to take our innovations to the marketplace, which is a lot about what we're going to hear about today. Not only are they the only ones on the move, in March, as some of you saw, the opening of the new home for the Keenan Center for Entrepreneurship. The Student Entrepreneur Center across from the Ohio Union is designated for students by students. This dedicated space provides tailored resources and mentorships for our student innovators in an inviting space. And if you don't know where that is, if you remember where the old Eddie George's is, yes? More familiar? Yes, it's great if you have a chance to check it out. It's also available if you need space, and thank you for the teams that really made that happen. Ohio State startups have also had a stellar year, as some of you may know. Coloma, out of the College of Arts and Sciences, a geologic hydrogen startup from last year's Innovation of the Year, Innovator of the Year, Tom Dara, has raised over $300 million. So we won't take credit for that, but he was our winner last year, so no pressure on the winner this year. Some of you know Coloma. Coloma will be having residents in this building, which is really exciting to keep that work here. I want to recognize Dean Susan Olisic in the back that really has made Coloma be able to come to life with Dr. Dara and his team. Coloma is working to extract gas from underground geologic deposits and turn that into clean energy. Dr. Dara's technology started in his lab in the basement of Mendenhall Lab, but soon his company will be leasing space in this building, as I mentioned, to continue the great work. Another Ohio State startup success out of medicine comes from Dr. Shahid Nimji, a neurosurgeon, a previous Research and Innovation Spotlight speaker, and early career Innovator of the Year finalist. And so again, we won't take credit for that, but it's likely to say it wouldn't have happened without winning that award. Basking Biosciences recently closed on $55 million in financing as it works to develop a drug capable of resolving blood clots to reopen blocked arteries and treat strokes. And one of the really best example of this aptamer technology moving forward for the treatment of cardiothoracic disease. And those aren't the only impressive numbers we've seen. In June, our research and creative expression community closed out fiscal year 23 with over $1.4 billion in research expenditures, a new Ohio State record. Then in November, the National Science Foundation released its annual higher education and research development rankings, and Ohio State moved up to number 11 in the country among all institutions, and now has surpassed Harvard as well to be in the top and near the top 10. While these numbers are impressive, and we talk a lot about numbers and rankings at Ohio State a lot, the foundation of what we do every day is science and creative expression. And that's why we all became scientists. That's why we all spent a lot of time in graduate school, a lot of time nights and weekends, the staff that really make this happen. That's why we're here today, and that's what we're here to celebrate. From amazing progress in gene and cell therapy research to CFAES researchers working to develop plants that can grow in space and harsh environments on Earth, to the development of battery cells to move technologies from the lab to the electric vehicle market, to helping Intel in the semiconductor industry grow in Ohio. Ohio State and Eric, and many of you in the room, all of our colleges are supporting the research and innovations that are literally changing the world each and every day. We've also supported the opening of the Center for Software Innovation and helped to host the first Startup Midwest Summit, attended by over 200 people in the software and the venture development department. In our partnership with Techstars, just recently launched its first accelerated cohort, which is over here in Mount Hall. We have 5 companies from the Columbus area, others hailing from the Bay Area, New York City, Los Angeles, Chicago, Florida, New Jersey, as well as London, Berlin, Israel, and even Armenia. I want to acknowledge the members of the team who may not get the headlines, but without all of you, none of these successes are possible. Thank you to the members from our sponsored programs team, research compliance, secure research, EULAR, our team that supports the critically important review boards and the IT systems. And I want to express our thanks to all the staff across all the colleges that are here today. We would not be here today without you. None of these successes would happen without you. Thank you to everyone here who has supported you, supported us, and we want to support you. So we're now here today to listen to some science. I'm excited to hear from our spotlight speakers who will talk about their research and innovations. The ERIC team is so proud to support them, as well as our deans and their colleges and colleagues to succeed, support, and amplify their efforts. First, we'd like to take the opportunity to introduce Xia Ning. Dr. Ning is the definition of interdisciplinary as a professor in the departments of biomedical informatics and computer science and engineering. She also holds a courtesy appointment as a professor in the College of Pharmacy. Dr. Ning was awarded seed funding through the President's Research Excellence Program, administered by our own Dorota Brzezinska and her team. Dr. Ning's collaborative research looks at using AI to create better, more targeted medicine. She has already taken pre-seed funding and received significant additional grants from the National Institutes of Health. Dr. Ning.
Well, thank you. Thank you for having me here. So artificial intelligence. So nowadays, this is a topic of intense interest. So let me ask you a question first. How many of you have used ChatGPT to generate new tasks? Just raise your hands. Great, a lot of hands raised. So as you know, ChatGPT is a type of generative AI or Gen AI, which can generate new content. So in my research, I ask a very big question, like, can we use generative AI to address the most fundamental questions in science and humanity? So take drug discovery as an example. So this process has been not just slow and expensive, with an extremely low success rate. So it takes about $1 billion and more than 1 decade to bring just one single successful drug to the market. So can we revolutionize this process using generative AI? So traditionally, chemists just keep looking for molecules and see if they can perform as a drug. So this process is going to be painstakingly slow because they have to sift through millions of molecules. So imagine instead if we can magically create the molecule that we really need. So that's why I'm here to share with you my journey about don't look, just create. So in my research, we developed the most advanced generative AI methodologies to create small molecules and their 3D structures such that they can bind to a protein target of interest. So our method can facilitate the generation of atoms and their precise locations within the binding pocket. And we also generate the bonds that connect these atoms together. So this is a generation process from our Gen AI technology. So in this particular example, a molecule is generated to bind to an enzyme produced by CDK6 gene. So this gene is closely related to multiple cancers such as leukemia, lymphoma, or carcinoma. So molecules that bind to this gene exhibit very significant therapeutic promise as CDK6 inhibitors and potentially offer treatments for these cancers. So it takes our generating method about a second to generate this molecule, plus another 3 days to train the generative AI model. So compared to the traditional method, which can take months, our method offers a significant reduction in time and resources, potentially revolutionizing how drugs are developed. So this is the molecule that we generated, and compared to the molecule that is approved by FDA as a drug for this gene. So our molecule actually exhibit properties that are closely resembling those as you observed from FDA-approved drugs. So that means our method has a good potential to produce very promising medication. But as you see here, our molecule is significantly simpler, indicating that it could be easier to synthesize. But more importantly, our molecule can bind to CDK6 with even higher binding affinity, indicating it could have even better efficacy. So you may wonder, oh, it's generated, but can you really make them real, or they just exist in the AI fiction? So actually, typically, chemists will look for some ways to synthesize this molecule, and they always have a difficulty to find the best molecule or the best way to synthesize the molecule. But given generative AI, here we have a different solution. So let's take a look at this molecule, mavacamten, which is approved by FDA for treating hypertrophic cardiomyopathy. So our generative AI method can accurately generate the patented synthetic reaction for making this molecule. But at the same time, we can offer you more options which are also viable. That means we can give you more opportunities to really simplify and expedite the synthesis process. So in this example, we use 2 smaller molecules to make the final molecule. But you may wonder, what happens if even these 2 smaller molecules don't exist? So in this case, we have to devise a sequence of reactions to make all the intermediate molecules until we get to the final molecule. So one way to do that is we start from all the commercially available molecules and try different configurations until we get there. But our chemists suggest a different solution or a different strategy. So that is, we start from the final molecule that we want to make, and we look at which bonds are breakable during reactions, and we go from there and work backward. So given this piece of information, we were able to come up with a totally different and totally new idea on how generative AI can do the job compared to anything existing in the literature. So that's why all the fruition of my research owes everything to such interdisciplinary collaboration. So in my team, we have AI experts who leads the development of the most advanced AI technologies. But we have chemists who give us the chemical knowledge to guide such development. And we have data scientists who give us the insights gleaned from the data. So by combining different ideas and different methods, we can really speed up the innovation. In the future, our aspiration is to develop generative AI method which can design personalized drugs for each individual patient. And hopefully those drugs can be manufactured efficiently and economically so that it can be used to cure the disease. And our endeavor is to develop generative AI as a trustworthy tool such that they can help us foster more scientific innovations and enhance the well-being of humanity. Thank you.
As those structures were flashing in front of our eyes, I was just wishing we had this available when we were all taking organic chemistry. Nope. So next we will hear from Caitlin O'Brien. Caitlin is a member of the second cohort for the President's Buckeye Accelerated Program administered by Eric's Keenan Center for Entrepreneurship. The Accelerator supports student founders as they prepare a startup for launch with mentorship and funding in a structured year-long program. As an aside, I invite you to join us on May 1st as we await and award the third cohort for this program. Caitlin is a founder and director of Solstice Outreach, which provides interactive STEM programming for K-12 students through portable planetarium shows. She also recently graduated from Ohio State with a degree in astronomy and physics and a minor in science and engineering in the public interest. Caitlin.
So coming from the astronomy side of things, entrepreneurship isn't something that I expected to have a lot of opinions about. I can pinpoint the exact moment that I decided that I was going to be going into STEM and specifically astronomy as a career. I was at an observatory at a local outreach event where students and astronomers talked to the public about the constellations and pointed out the stars. And I had been interested in space before then, but I had never considered it to be a viable career option. After a particularly long conversation with a graduate student about his path to the stars, I went home and I planned out the next 10 years of my life to become an astronomer, a path that with some deviations I am still following to this day, all because someone showed me the stars and told me that I could. I was incredibly lucky to have that experience, and unfortunately not everyone does. That ripple effect that I experience is what I founded Solstice Outreach around. Who here has been in a planetarium before? Wow, that's a lot. I want you to think about where and when you went, what you were experiencing, what you were doing, and then what you saw and how it made you feel. Solstice takes that experience and brings it anywhere. We give interactive, engaging STEM outreach directly to communities through these portable planetarium shows. The field of space science has always drawn me for its ability to bring everyone together, and that's really what we're trying to do there. Everyone thinks that space is cool. That's evident by how many people here have been in a planetarium before. But not everyone thinks that space is relevant to them. Not everyone thinks that there is a space for them in space. And by bringing a space science directly to these communities that might otherwise never experience it, we connect the idea of this is cool to this is possible. Solstice is founded around this idea that if you can see it, you can be it. We bring the portable planetarium directly to these communities that might otherwise be out of reach, aiming specifically at younger audiences that haven't yet decided what is and isn't possible. We show them the stars, and more importantly, we show them someone that looks kind of like them, a planetarium presenter that is an undergraduate student at Ohio State who might just be 5-10 years ahead of them in their career path. And through that, we're able to be that spark of inspiration. We're able to be that ripple effect. This is the pitch that we brought to the President's Buckeye Accelerator last April. We were one of 51 student ventures competing, and we made it through an initial round of pitches to the finale, which is what you see in these images here. Ultimately, we were selected for $50,000 in funding through the President's Buckeye Accelerator Prize, and this is what enabled us to purchase our first portable planetarium system. One of the main points of our pitch was the idea of space as a force for good, combining my background in astronomy and planetarium presenting with entrepreneurship. As a student startup, you hear quite a lot about corporate buzzwords like total addressable market and minimum viable product. But from the beginning, we knew that our real selling point wasn't our addressable market, it was our total inspirable market. Earlier, I asked you if you've been in a planetarium. This right here is a planetarium. That gray duffel bag on the left becomes this 5-meter wide big blue dome on the right. And those of you that are familiar with the Student Entrepreneurship Center will recognize that this is set up in that center. Once inflated, we can fit 25 adults or 40 young children inside. It can be set up in a gym, a cafeteria, an auditorium, even a large classroom or a conference room can suddenly become a planetarium. Once inside, students can ask questions. Everything is interactive. The students can talk directly with the planetarium presenter and guide their own show. Everything we do is based on who is in the dome. It's not a pre-recorded movie, it is a presentation that is given by an Ohio State student that is passionate about what they're talking about. If we have a student that really, really wants to see Mars, we can take them to Mars. Everything that they want to see is a possibility. Now, we had our first show ever last week. These are photos from those shows that we gave. And when you're doing something like this, when you're doing something this big, and especially the backing of the PBA prize, there's always this little voice in the back of your head that says, what if this doesn't work? We know that space is cool. We theoretically think that if you put kids in front of a bunch of space, they'll think it's interesting. But we don't know that until we do it. And so this bottom right corner is from our very, very first show, and the first few seconds of it, in fact. This is before we showed them anything. You can see that there's still a blue sky and clouds. There's no stars, there's no planets. We had not left the Earth yet. And you can see their faces. The moment that we brought them into this dome, they were transported to another reality. We had kids asking so many questions that we all but gave up on the planned programming that we had expected to be doing, and instead just interacted with them. They asked us questions, and we showed them the answer on the dome. I had one student that asked us if we would have enough time to see the heliosphere, and he was in 2nd grade. I had another student who said, I am so excited about this because I want to study the sun. And I told him, that's called being a heliophysicist, and it is a very good idea. In one of the shows that I was presenting for, in the middle of our conversation about Jupiter or wherever we were, I had one of these students stand up from where they were sitting, run up to me, and just wrap me in this big hug. He said nothing. He didn't explain. He just hugged me and then went and sat back down and continued watching the show. And that was the moment that I really realized that what we were doing was going to work. And it was actually having an impact on these students. And we were just getting started. If any of you here think that you know a group that might be interested in one of these planetarium shows, I implore you to reach out to me after this or with the links that I provided on this slide. Everything that we're doing is in pursuit of this idea of making the universe available to anyone, anytime, anywhere. We believe that access to STEM increases the possibilities, and Solstice increases access to STEM. Kids from underserved communities often don't believe that STEM is something that they're able to be good at, never mind something they can make into a career. But we know that that one moment can spark a lifetime of inspiration, a lifetime of curiosity. And we know that because it happened to me, and I believe that that can happen to anyone. Thank you.
So one of the things that we're continuing to work on with our ecosystem here is not only to be great mentors of incredible, incredible talent, but also think about things like being first customers. So if I could get everyone to pull out their phone and take a picture of the contact information, would challenge all of you to let's figure out a way to get this to work and work well. And so please take an opportunity, and if you can think of classrooms, you can think of your own organizations, let's really make sure that this cranks. So thank you and congratulations for your work. it takes a team, and I know there are a lot of people on your team, and so if there's other members of the team here, if you can raise your hand so we can acknowledge you. If not,
They are in the pictures on there. They weren't able to come tonight, but
understood. They're probably all working and recovering from yesterday or the day before. So thank you again, Caitlin. And last we have today Dr. Richard Gumina. Dr. Gumina is Associate Professor of Internal Medicine, wears many hats, mentor, leader, scientist, friend. He serves as Associate Division Director currently for research in the Division of Cardiovascular Medicine. Let's hear from him today on how his interdisciplinary approach is exploring innovations that are unraveling the factors that contribute to cardiovascular disease, still the #1 cause of death in the United States. His focus on innovation is what we are driving to support more of through the work of the innovation and commercialization teams led by Paco Herson and Kevin Taylor and many others. Dr. Gumina.
Thank you, Dr. Moeller. So my name is Rich Gumina. I'm an interventional cardiologist. I work at the Ross Heart Hospital, and I take care of patients who have heart attacks. How many of you know someone who's had a heart attack? How many of you might have had a heart attack? Okay, good. No hands. Thank you. How many of you are having chest pain? I am. But I want to share with you my translational research that we've done, getting from bench to bedside and back to bench again. So my life is usually a blaring alarm at 2 a.m. in the morning, or 2:30 in this case, that goes off. It always bothers my wife, bless her soul, she deals with me, to wake me up to say that someone is on the other line with a patient who's having a heart attack. And so in this case, it was actually a physician who was at an outside hospital having a heart attack, and this is the ECG that we're looking at. And if you look towards these red arrows, what you'll see is that there's elevation of that segment, kind of down to the left, your right side. You'll see that it's flat after those little upticks, and it's significantly elevated. And that's an ST elevation myocardial infarction. This is a medical emergency. These patients have to be brought to the cath lab immediately. And so a call goes out, we all get another page, and we've got about 30 minutes to get into the cath lab to try and save this patient's life. Once we get them there, we thread a catheter up their arm, and then we do an angiogram. And in this case, when you're looking from left to right, there should be an arc coming down of a vessel that's missing in this case. And you see a little of that x-ray dye that's hanging up there. That's the blocked vessel. That's the left anterior descending artery. This is known as the widowmaker. When this vessel goes down, people generally don't make it. So what we do is we thread a wire into this vessel, and we get the artery open, and you can see now that arc is there. That vessel is coming down across. And then we balloon that area. We also take a stent, and we solidify opening that artery so that stent opens it up. And you can see now you've got a pretty robust flow into that vessel. So we pat ourselves on the back. Great job. We saved them. But this is not the end of it, because these patients are incredibly sick at this point, and they can go into cardiogenic shock or have other problems. So this is what I study, this translational medicine, studying this fork in the road. Patients can go on to having arrhythmias, heart failure, or even death, or they do incredibly well. They recover, return to normalcy, and you wouldn't know that they've had a heart attack. And in this case, our patient was really sick. So we had to put other support devices in to help them, to get them through this heart attack, and then work on medications to try and get their heart to recover. And this is what we study. Trying to understand this translational medicine of how do we understand what's going on with the patient, and then how do we study it. So we do basic science work where we take observations and models that we try to mimic human conditions with, and then we translate that to humans, see if they resemble what's going on, and then what we do is see if we can translate it to patients. And so it's a whole process of lots of teams that work together. And so I just get to be the spokesperson, but there's probably 30 people that are in the background that do all of this, including our nursing staff, our cath lab staff, and then all the technical people that do the wonderful work. So when someone has a heart attack, what happens is the heart muscles, the myocytes, get damaged. And then there's inflammation that occurs. And this inflammation is what we study. And specifically what we study are nucleotides that get released when the cells die off. They get released by platelets, which cause the heart attack. And these ATP and ADP molecules that get released can signal and bring in more immune cells that then will cause more inflammation and more damage. They also signal through other receptors, and they cause other recruitment and damage to the vessels, damage to the heart muscle. But there's a general that can come in, this T regulatory cell, that contains enzymes, CD39 and CD73. And this general cell that comes in can guide things either to a good place or to a bad place. And so if it has low levels of CD39, we think that it can't get rid of the ATP and ADP. If it has high levels, though, it can generate adenosine, which then has its own receptors that we think are beneficial. And so by driving this, by having the right generals in the place to drive this process, we think we can affect the outcomes in myocardial infarction. So how do we get there? Well, SNPs, or single nucleotide polymorphisms, make us all different. And so we have 2 chromosomes. Just a single base pair can be different and cause our differences. So when you look around the room, it's just all these little subtle differences that make us who we are. But you can have a heterozygous like this, where it's 1G, 1A in that spot, or you can have homozygosity, so both AA or both GG. And usually it's these homozygosities that can cause disease or give us indications of how it can affect disease. And so in our case, when we look at this CD39 and the T regulatory cells, what we see is that if you have 2 A's, you have high levels of CD39. So the generals that come in have high levels. If you have 2 G's, though, you have low levels. And so those generals aren't as well equipped, we think. Now, you say, well, how many people really have this? So 30% of you have AA, 20% of you have GG, and 50% are in the middle. So it's not a rare polymorphism. It's pretty common. So what we're studying is looking now in patients who have heart attacks to try and understand if you have that AA genotype or that GG genotype, does the inflammation that occurs after you have a heart attack resolve quickly, or does it get prolonged? And so what we did is we did a study with a team that was in Denmark, took their patient base, they had already had genetic information on them, and we looked at whether or not they have differences based on genotype. And what we found was that, in fact, if you have high risk with this GG genotype, so low CD39, your outcomes were worse. And if you have the low risk phenotype, this AA phenotype, then your outcomes were better. I can't show you the actual graph because we submitted it for publication and we're waiting to get that published, so I don't want it to go out before the publication. But what we're working on now is taking those high-risk patients, like the one I presented to you, and how do we drive them into that low-risk category by either understanding, are they high risk based on their genetics, or developing novel therapeutics so that we can improve their outcomes. And so that's what our whole team focuses upon, and hopefully what you'll hear next time when Peter presents is that we've developed a drug, because now we have people who can develop specific targets to this to make this all happen. So thank you.
I guess my understanding from listening to the first talk is you all could figure it out in about a minute and a half. So maybe we check back next week, Rich. So thanks again. So this afternoon, to get down to business, we're here to award the university's Innovators of the Year. These awards annually recognize 3 researchers, each at different points in their career, who are actively working to translate university discoveries through commercialization where their work can truly impact the community. I'd like to now ask Deborah Hernandez and Shireen Agarwal to come to the podium for the first announcement.
Hello, I'm Deborah Hernandez, Associate Vice President for Strategic Research Development, and Eric.
And I'm Shireen Agarwal, the Executive Director for the Center for Software Innovation.
We are presenting the Next Generation Innovator of the Year Award. This award is granted to an undergraduate student, graduate student, or a postdoc to recognize innovation and entrepreneurship that contributes to the development of a new technology. Let's hear from each of our finalists in their own words about their work.
My research is focused on triple-negative breast cancer, which is one of the most aggressive forms of breast cancer in women. What makes this most aggressive is the ability of cancer cells to migrate from one part of the body to another part through blood vessels which act as transport highways. Preliminary research from our lab has demonstrated that weak induced electric fields are a relatively effective treatment strategy compared to conventional treatment strategies like chemotherapy, owing to its less invasive nature and potentially less side effects. My research is focused on investigating the effect of weak induced electric fields on the permeability of blood vessels in the context of breast cancer. I study these by developing microfluidic devices which have the form factor of a flash drive, and these devices house different cellular components and extracellular matrix components, and all these components continuously experience fluid flow through a novel methodology that does not require any power source and solely relies on mechanical design to pump fluid for several hours to mimic the blood flow inside human body. Using microfluidics to solve healthcare problems is what I enjoy learning about and executing.
My research is about developing unique wearable or implantable biochemical sensor that's specific for human subject or animal. So one of my research is about a wearable type of sensor which can directly detect glucose inside the sweat. So this targeting the diabetes patient. So we collaborate with physicians in OSU Medical Center to study whether there's a real-time relationship between glutamate in the sweat and also in the blood glucose. My family members have diabetes and they need to take finger stick blood every several days. Since it's a more invasive method and it's not that user-friendly, so I think that's the reason why I think wearable is another step in the near future.
For this work, we've developed a new class of cell-penetrating peptides, and we're hoping that we're able to bridge a gap in modern therapeutics. Modern therapeutics basically have two broad classes. They have small molecules that go into cells and do their business there. And there are large biologics that stay outside of cells and do their business outside of cells. Our work in this lab for cell-penetrating peptides is trying to bring those large things into cells to effectively target what is about 75% of current diseases that are undruggable. We have successfully designed this new class of molecules. The Pay Lab has been working in cell-penetrating peptides for at least the last 10 years. Where this technology comes in is that it's a new form of cyclization for those molecules. We have shown some of those molecules to be pretty good. We are still working on optimizing them and working on what disease targets that we can actually drug with these new molecules. I've been fascinated for a long time on how to get into doing the work to be able to drug those undruggable diseases. And I was very fortunate to be able to find the Pay Lab and be able to come here and work with such great faculty and graduate students at Ohio State.
We are in the field of computational medicinal chemistry, so we combine computational structural biology and medicinal chemistry knowledge for small molecule and drug design projects to empower the process to make things faster and more efficient. What I do is I integrate computational tools with machine learning technologies and biophysics principles to know more about the structure of the protein target in order for us to design a better binder or better small molecule therapeutics to interfere with the targets of certain disease we're interested in. We do molecular dynamic simulations, and we also have a self-developed toolkit for explore the allostery and multiple conformational landscape for the protein targets or target of interest for the disease. And in the process, we are going to use the established tools like molecular dynamic simulations and existing structure prediction tools that are available to everybody. And we're going to integrate those tools and come up with new methods in order to know more about the allosteric pathway and protein conformation landscape of the target we're interested in. We already saw the emerging publications or results on machine learning, artificial intelligence in drug discovery. And I want to set my foot in the field as well. I think they can be very promising in the future.
Our next generation investigators are truly the future of research and innovation, and their passion and energy is contagious. This is my Oscars moment. Here we go. The 2024 Next Generation Innovator of the Year is Sam Sheng from the College of Pharmacy. Congratulations, Sam. Please come up to receive your reward.
I would say, even though my name is engraved in here, even though this is like for me, but this is definitely not a personal award. This is a team effort. And today's recognition, there are, I think this recognition owe a huge part for many people in or not in the audience. And I have to mention them today. I would say this is a great team effort. Without the OSU research community, there's no way for me to get such achievements. And also in our college, what I admire more is this freedom and great research environment for me to fulfill my dream and ideas on the research. So I would like to mention Professor Nadine Krotz as our Dean in College of Pharmacy, and also Dr. Jim Fuchs as our Associate Dean in the Pharmacy College. And also all the faculties, I saw there are many faculties from our college. Our college become a better place because of you, and I really appreciate that. And other than this, I also want to take this opportunity to thank my advisor, Dr. Tom Lee and Dr. Xiaoling Chen in the audience. I know Dr. Tom Lee for a long time. At that time, I was still a very naive, stupid undergrad student. I was broken glassware every day. Like, Dr. Lee was still bearing with me, and I really appreciate that. And from Dr. Lee and Dr. Chen, the only thing you get from them is encouragement, is inspirations, is motivations, and opportunity to try different things and to fail. I think their tolerance to me is the most invaluable thing I can get from this college and from our research teams. I think this is very, very critical. I really appreciate that. And they always educate me about the nature of interdisciplinarity of medicinal chemistry or computational medicinal chemistry. If you want to design drugs, we cannot just be chemistry students. You cannot just learn computation. You need to know more about the language from other peoples. And that's the reason why we get these several collaborations with the best faculty and collaborators in our college. I would like to mention that I have a chance to sit in Dr. Chris Koss' lab meeting to learn pharmacology kind of things in my career path. And we also have the collaboration with Dr. Ross LaRue and Dr. Amit Sharma together to study antiviral drugs and also peptidomimetics and peptide drugs. Those are extremely valuable to me. And I also want to thank Dr. Men Wu and Dr. Zhihong Lin for their helps during my enzyme assay experiments. I think those are extremely critical for my research experiments. And also my committee member who may not be here today, Dr. Carl Werbers and Dr. Mark Mittenfry in our division, who has been my committee member, having meetings every years, and we have been discussed a lot during this career. And there are many, many people I want to thank, and I think this is a great honor, then this owe a lot parts to them. And I also saw my lab mates are here from Dr. Chen's lab. I enjoy the time spending the time with you. And most critically, I want to also give the credits to my girlfriend, my sweetheart. I love you so much. Wish you my life becoming happier. So this will owe a huge part to you as well. Thank you so much. Thanks for this award.
Okay, that's a lot to follow. Big congrats to you, Sam. I'm Cynthia Karn, Senior Associate Vice President for Research Operations, and Eric.
And I'm Kevin Taylor, Chief Innovation Officer for ERIC.
Our final two awards recognize researchers who are more established in their careers. These researchers are actively working to advance discoveries through commercialization activity by disclosing inventions, receiving patents, licensing technologies, or forming startup companies. These activities are important to not only improving lives, but to supporting our land grant mission. Let's hear first from each of our Early Career Innovator of the Year finalists.
The research program that we're building in our lab focuses on understanding the cellular interactions in the tumor microenvironment of a very malignant form and a very aggressive form of brain cancer called glioblastoma. What we are trying to do is understand how these different cells talk to each other and how it affects the immune responses in glioblastoma. And what we found specifically is that neurons, which are an integral component of the glioblastoma tumor microenvironment, can talk directly to the immune cells. And we're trying to understand that if we block or augment this dialogue between these two cell types, we can enhance the anti-tumor immune responses. Right now, we have identified neuropeptides, so specifically some sort of chemical which these neurons make, which can affect the immune responses directly. And we see that targeting this neuropeptide can improve anti-tumor immune responses in context of glioblastoma. I have always been very, very fascinated by how the immune system can be leveraged to fight cancer. So immunotherapy, it's my favorite subject, and this field has been evolving very rapidly. So I've always been fascinated by that and want to expand this therapy in broader range of cancer types and improve the life of patients.
We made 3 discoveries. First, organoid-3 also cleave RNAs. Second, organoid-3 splicing activity is drastically increased when they are loaded with the 14 nucleotide cleavage-inducing tiny RNAs, which we named ctRNAs. Lastly, we also devised a system to load a ctRNA into the endogenous organoid protein in the cell. I am a co-founder of a company that is licensed to use the Ohio State University intellectual property and funded by top-tier is venture capital firm. So the goal of this company is to develop ctRNA-based therapeutics for diseases whose the target have been undruggable by conventional approaches. So far, to the best of my knowledge, only 10 papers about the tiny RNAs, almost nothing. So I want to make a field of the tiny RNA. Nobody knows what they're doing in our body. So it's exciting to start this project.
My research here at Ohio State consists on developing microtechnologies, biomaterials, and molecular imaging techniques for the characterization of circulating biomarkers with the idea of better diagnose diseases, specifically in terms of cancer and infectious diseases. Through this improvement in technology, provide a much more accurate way, for example, how these diseases are diagnosed. For example, it could be also much more rapid technology that is used in the semiconductor industry to create these microfluidic devices. And these microfluidic devices allowed us to process biofluids that came directly from patients, such that we can identify these circulating biomarkers that can have a better way how to diagnose the disease. If the disease is diagnosed much more early, like in the terms of cancer, is actually very important. So this is the idea on mind for us, such that it would provide an additional value for these patients, such knowing that this disease can be detected at a much earlier stage. And that's actually a great way for me as an engineer applying my curiosity and ingenuity in problems that are related to medicine. So that's a great opportunity for me.
It's exciting to see the amazing work going on with our early career faculty. So let me open the envelope. All right. I'd like to congratulate Dr. Kotaro Naganishi from the College of Arts and Sciences as a 2024 Early Career Innovator of the Year winner.
There you go. Congratulations.
First, I really appreciate the leadership team selecting me. It's a great honor for me. Then I'm grateful for Chris Wilson, our senior is the licensing officer of the TCO, and is the Nicole Snyder. Yeah, is the director of the venture, is the development. So for your very thoughtful is the support and hospitality. Okay, so then is the also is the Janelle is Cleveland. Yeah, so you're fantastic as a patent lawyer. Without you, we couldn't patent 3 new patents on April 1st. So I know we have more. So let's keep going on. So, okay. So then I also want to express to my home is the department, chemistry and biochemistry. So I joined Ohio State when Susan Oresic, so you're the chair. Since then, the department has been very supportive to bring more money from NIH, right? So then I also say thanks my lab members because they tested tons of my crazy ideas. Okay, I appreciate. The last 3, so that I want to thank my daughter Sophia and my wife Cherokee. Yeah, so you are my sunshine and so the energy source, especially the Cherokee. So you have been supporting my science career. So ever since before we moved to the United States. Thanks so much. Also, thanks for your pay attention. Thank you.
Okay, that was definitely worth it. Congratulations again. So now for our final award, the creation of a separate award categories for our faculty and staff with more established research programs allows for cultivation of an entrepreneurial spirit among all of our investigators. Let's listen to each of our impressive Innovator of the Year finalists, and then following that, our Vice President for the Knowledge Enterprise, Dorota Brzezinska, will come up and present the award. Thank you.
My basic research focuses on neuroregulation of metabolism and cancer. We aim to understand how an individual's physical and social environment, as well as lifestyle factors, influence the central nervous system. We developed 2 AAV platforms. One is to target the brain with a built-in safety control, regulate therapeutic gene. Another platform is to detarget liver, where target adipose tissue, which can potentially deliver many genes systemically at a safe and cost-effective dose. They will enable gene therapies for mainstream disease with a larger patient population. We can potentially treat genetic form of obesity and lipodystrophy. With my research progress, I was inspired by patients with genetic form of obesity, children with Prader-Willi syndrome and their families. We definitely feel the urgent need. We want to help to find a cure.
If you look at traditional IT controls, they're about blocking people, blocking actions, and locking down, say, devices and networks. When we start building perimeters around data, it's almost always the more difficult approach, and just in the name of protecting data, building perimeter around the data doesn't work well. So instead, the idea that we had was to bake protection directly into the data so that security can travel with the data. That gives freedom to organizations, businesses, anybody who is dealing with data. Because your security travels with your data, you don't have to worry about what happens to it if it's stolen. So as a result, data theft becomes irrelevant. The true innovation comes not when you necessarily solve a complicated problem. The true innovation comes when you find a simpler way of solving a difficult problem.
My research is in the biomanufacturing, using synthetic biology to engineer microbial strain for fuel and chemical production. We have been working on a number of products and using my technology. The first commercial production of my technology produced product is the calcium propionic acid, which is a nature preservative used in bakery product and also in some other food product, in dairy and so forth. It is fermentation produced from sugar. Since my college, we were very interested in the microorganism, their ability to make wine, food, medicine, and everything pretty much. So that is really a wonder of the world. Okay, so we wanted to do the microbial fermentation to produce not only food, drink, but also useful medicine and today chemical or almost everything you use in your daily life.
What an impressive group of innovators, and we are really truly lucky to have them all at Ohio State. And it's my greatest pleasure to announce the 2024 Ohio State Innovator of the Year. And the Oscar goes to Dr. Emre Koksel. Could you please come to the podium?
Thank you very much. Thanks. I appreciate it. Thank you very much for this award. I'm truly humbled. First, I would like to thank my family, especially my wife, Ishil. She's here, and she's been the anchor to my foundation, and none of this would have been possible without her. So, secondly, as a professor, I think the highlight of the whole career is working with students. I mean, when you work with young people and try to figure things out with them, that's the most amazing experience, and that's kind of been the highlight of my career, and I'm really thankful to have the greatest students here at Ohio State. Obviously, we don't know it all. When we are taking the ideas and building businesses out of it, the sooner that you acknowledge that you don't know everything, the better it's going to be. To that end, the mentors play a great role. I mean, you have to surround yourself with people who know what they're doing, who are like world-class in the stuff that they're doing. So the sooner we do that, the sooner we realize that, that's the best, that could happen to us. And we are also fortunate to have, the best mentors here in this ecosystem. So I'm really thankful to have them around me. So lastly, I'm thankful to the whole ecosystem here. So let me explain. When you think about innovation, you think about these videos are focusing on the science, the math, the nice software, the nice hardware and everything. That's the basic. But unless you take the next step to touch the human component of it, it doesn't mean anything. I mean, you have to genuinely understand the needs of the customers to build a business out of your idea. And I feel like that's the distinct advantage that we have here, not just at Ohio State, but the region, right? I mean, the region has in its culture the humbleness, the down-to-earth nature. We have empathy. And, in my opinion, when we are building the next, for example, the Silicon Heartland, we should put it at the center, like the human component as the strength of this region. And I'm really thankful that, everybody, the ecosystem, the people, has shared that, empathy with me. And, it's a great place to be building businesses, and we should make it our identity. So thank you very much, all of you.
what a great time to be at Ohio State. What a great time to be in science. What a great time to be in the community. Congratulations to all of our innovators of the year who are inspiring us to wonder more, inquire, and discover. And I just want to quickly, there are a lot of people in those videos that weren't up on stage. So if you were nominated, we'd like you to please stand to be acknowledged for all the great work you're doing, please. Just quickly, I also want to make sure we thank the people that spend significant amounts of time. You can sit. All the time that the mentors that took the extra hour, the extra couple hours to make sure you nominated your teams. And it says a lot about the ecosystem that we have that level of mentoring in Ohio State where people are willing to do that. So congratulations to everyone. Thank you all for each of you who have joined us this afternoon. No matter what role you play at Ohio State, each of you play an important part in ensuring research, innovation, creative expression, and knowledge continues to thrive here at Ohio State. And for that, we all thank you. On behalf of the entire team who put this event together, and thank you for doing this, want to thank you all for being here, making the ecosystem what it is today. Please stay and enjoy the reception, which is just outside of you. Safe travels, and thanks for being here tonight. Thanks again.